Let's get into it then. We've come across metabolism before, so let's just look at enzymes and see what exactly we're talking about when we're talking about enzymes. First of all, if you're asked what's an enzyme, all you need to say is that there are biological catalysts. Now, you need to understand what the word catalysts are, and you could well be asked to explain what is that? What is meant by a catalyst? But if you're asked for a definition for an enzyme's biological catalyst, is perfectly fine, full marks. What catalysts are, if you're a chemist, you'll know this. And if you might remember back to junior science, the catalyst that you would have come across, apart from amylase, the catalyst that you come across that isn't a biological one might be manganese dioxide. Now, the reason that might ring a bell is because what that does is it speeds up the breakdown of hydrogen peroxide. And you would have done that in the chemistry lab as a junior. Well, maybe you might have done it depending on what the restrictions were like in order to produce oxygen gas, right? So it's the preparation of oxygen gas. You use this stuff, manganese dioxide. It's a catalyst. Now, there's no way you could put manganese dioxide near any living things, really. And it's not going to, you can't put it, it's not going to work inside cells. In fact, it's too toxic. It's going to destroy cells. So not all catalysts are enzymes, but all enzymes are catalysts, right? So lots of catalysts are horrible chemicals that you wouldn't put anywhere near living things. Those that are produced by living things and used in living things, we call them enzymes, okay? They are almost all proteins. And in fact, if you're asked in the Leaving Cert, what kind of biomolecules are enzymes made of, you're going to say proteins, right? But it turns out, and we'll see later on in the year when we come to protein synthesis, that ribosomes, and ribosomes from a couple of weeks ago, they're the sites of protein synthesis. And they're made of RNA and they're made of protein. And for a long time, it was thought that the protein part was the enzyme. Turns out, no. Turns out that the RNA is the enzyme that catalyzes the chemical reactions, the synthesis of proteins at the ribosome. So almost all enzymes are proteins. Let's put it that way. So what does that mean? Well, we've come across this before, right? Proteins are chains of amino acids. And when we left biomolecules, we knew they're, polypeptide chains that are folded into a particular shape that give the protein its that allow the protein to function. But it's all about the folded shape. Okay, so folded shape is going to be your key words here. So the correct folded shape is what gives an enzyme its unique activity, right? And when I say unique, usually it only catalyzes one reaction, right? That's the whole the usual story with enzymes, and we call that correctly folded shape. We call it its native shape. Okay. So under the right conditions, then enzymes should have this particular folded, unique folded shape, which gives it its function. And we'll see how that works in a minute. Okay, so apart from them being catalysts and them being made of protein, what they do is they work by increasing the rate of metabolic reactions. That's what catalysts do. So catalysts generally speed up, but I suppose technically you could say catalysts change the rate of chemical reactions. But in the case of biological reactions, metabolic reactions, we're pretty much always talking about that they speed things up. They speed up reactions that, to be honest with you, otherwise, without the enzyme being present, then it would be way too slow or wouldn't happen at all. What we know is life. What we know is, metabolism and the way living things run just wouldn't work without these enzymes. So they're specific. They have a unique folded shape. Each enzyme acts on only one substrate. That's generally true. And it's the unique folded shape that causes that property, right? That results in it being able to bind to only one substrate. And by the way, the word substrate in chemistry in junior cycle, we would have, you would have talked about chemical reactions, and the reactants are the chemicals that react together. They're called reactants, and then you get the products, right? When we're talking about biological reactions, generally speaking, with enzymatic reactions, we talk about the reactants are what we call the substrate or substrates. right? So enzymes act on a substrate, and then you get a product. So you get substrate catalyzed by enzyme, giving you product. Okay. So what else can we say? Well, we can say that the rate of action or the rate of activity or the rate of action of enzymes is affected primarily by temperature and pH. Other things affected as well, like the concentration of the enzyme, the concentration of the substrate. There's things like there are inhibitors, enzyme inhibitors. We don't go into any of that stuff. You really just need to know that the rate of activity of enzyme is primarily affected by temperature, pH. We'll look at those, and you've got to do and probably have done already practical activities that investigate those things. So the last thing we'll say about the general properties is that if you many enzymes, but not all, are detrimentally affected by very high temperatures and by either very high or very low pHs. So very low temperature doesn't denature enzymes, right? What we mean by denature is that the activity is reduced, practically lost, and often irreversibly lost, right? So you can have reversible denaturation, but you can have mostly it's irreversible. So if you heat them to a very high temperature, enzymes generally become inactive. They're denatured. So when you cool them down again, they still don't work anymore. It's not true for low temperatures. So we store enzymes in the fridge or the freezer because at low temperatures they stop. They become inactive. But then when you take them out of the freezer and allow them to thaw again, they work perfectly fine. So it's a way of storing enzymes safely, because enzymes are fairly delicate generally. They're proteins, they tend to break down. So we store them in the freezer because low temperature, stabilizes them, right? High pH and low pH will, again, generally speaking, cause enzymes to denature. So we've said already what they do is they speed up the rate of chemical reactions, biochemical reactions, metabolic reactions in cells. So in that way, what they do is they control cell activities. Now, here's an interesting thing. So when we were looking at the cell structure, and we would have thought, tried to come up with what's the role of the nucleus. So the role of the nucleus is to control metabolic or cell activities. So how can enzymes also be responsible? Well, because it's in the nucleus that you have the DNA is stored. And by and large, genes, which are sections of DNA, they code for proteins. That's what genes do. Genes, by and large, code for a particular protein. A gene codes for a protein. And a lot of those proteins are enzymes. So now you can see why it is that we say that nuclei, or the nucleus, is the organelle in the cell that's responsible for controlling the activities of the cell, right? The metabolic activities. It's because largely what they do is they produce, or a lot of what they do, what those genes do, is they produce enzymes. Now, not all of them. There's some structural proteins that are needed. There's hormones that are needed. Proteins come in there's lots of different types, right? But in terms of controlling cell activities, enzymes are king. Just kill my tones here. So, as I said, a lot of these reactions just wouldn't take place unless there was an enzyme there to help it along. And if you remember from junior cycle, these activity curves. So this one shows you on the left, you got a chemical reaction that is occurring without enzyme, and this sort of hump that needs to be overcome. That's what we call the activation energy, right? It's the energy that's needed to get the reaction going. So what enzymes effectively do is they reduce that amount of energy that's required. And by and large, what we're talking about activation, and we're talking about heat. So the reason that enzymes are so important is because they allow chemical reactions in the cell to happen without very high temperatures. Right? That's the bottom line. That's usually what we're talking about, why enzymes are important. Now, here we go to substrate and product. Here's a whole rake of substrate, enzyme, and product combinations that you should know, right? Because most of them are digestive enzymes that you need to know about. In fact, they all are, right? Generally speaking. So starch is broken down by amylase to produce maltose. And maltose is a disaccharide, and that disaccharide, maltose, can be broken down by an enzyme, maltase. Most enzymes are named after the substrate. Amylase is weird, right? there's not, it's not starchase. But if you look at the other ones, my maltose is broken down by maltase, lipids are broken down by lipase, proteases break down protein, peptides, peptidases, and so on, right? That's the usual kind of way of naming them. But there's some odd ones. So old names we're stuck with, like trypsin and amylase.